Yes, I'm Rachel and I'm doing my PhD at Ghent University, and the title of my talk today is just on understanding and predicting fish movement in the vicinity of a barrier and fish pass, which is a bit of a mouthful, but it covers the whole thing. And today I'm going to first give a really quick background. I'll be repeating a lot of stuff we've already heard, but maybe you slept. And then go through like the first couple of chapters of my PhD thesis, and then ongoing work because I'm on a 4-year PhD. So yeah, background, as we've heard, there's a lot of freshwater fish that need to migrate in order to complete their life cycle. But as we've heard, there's loads of barriers fragmenting Europe's rivers, but what we also need to think about is that these barriers are likely to increase with the move towards green energy, for example, and that there's many systems where river fragmentation and the resulting impacts on fish have been underestimated. So there's a problem, it might get more and we might be underestimating it. And all of this has come together to cause a big decline in migratory freshwater fish populations. And as we've heard today, fish passes as maybe a solution to getting around a barrier. So that's a fish pass if you're not very familiar. But as we've also heard in Paul's talk, which was very good, is that they're not necessarily always going to be effective. This is just one graph from like one paper. But the passage success varies with traits of the individual species. So salmonids often have traits that help them pass successfully, but also with sight, with loads of other things. So it's not really a one-size-fits-all, and there's a need to really get to grips with what's going to be the best solution for a given site, a given problem. And yeah, that leads into kind of an interest in some groups, especially people who are really working in consulting with hydropower companies on like a way to be able to predict passage success before they go through the whole process of building a pass. Is what we want to do going to work? And for this, there's increasing interest in individual-based models. So these are also called agent-based models, like what James just spoke about. But I'm going to use IBMs because I feel like it. So for those who aren't very familiar with IBMs, as you can maybe guess from the name, individual-based models model individual elements of a system. So for example, a fish as it moves through the environment and tries to find a fish pass. And of course, you can model multiple fish. And that's one of the benefits of individual-based modeling approaches, is that you can scale up from the individual level and what the individual is doing to population level output. So in the context of fish passage, what are we interested in? Well, for example, passage success. So you can model the movement of lots of different fish and then look at how many of them manage to actually find the fish pass or actually enter it. And IBMs on the whole have been widely applied across ecology to loads of different questions, and they're very good at predicting into the future in novel environments, and you can transfer them between different sites fairly well. So the first chapter is a literature review on the application of individual-based models in the specific context of fish passage, as there's some well-known examples already. So what we were really looking for in the literature review was, to what extent have IBMs already been used to predict fine-scale fish movement in the context of freshwater fish and barriers and passage? For what species have they been developed? Because we've already heard about species-specific differences, so it's good to know. And how do each of these models tackle movement itself? So what parameters do they use to guide the movement? And what elements are potentially missing that could be included in the future? So initially I had 93 papers from a database search plus 2 papers that I just had on the side. Screened on the abstract and ended up with 16 papers because we wanted ones that, well, doing what was listed there. Did they develop an IBM for fish? And so on. And on reading the full papers, we ended up with 12 because, yeah, some of them actually didn't do an IBM or whatever. And what I'm going to note here is that while I say 12 papers, there was not 12 distinct individual-based models, as several of the papers kind of expanded upon previous models. And yeah, that's published. At the end I have a QR code you can scan if you're interested, because I obviously can't go into detail. So big thing first is the bias for salmonid species in the existing individual-based models. 9 out of the 12 papers detailed a model that could be applicable to a salmonid. there was a good mix of processes driving movement that really drew from what we know from literature and from like flume studies. So quickly, attraction rejection behavior from accelerating flows. Minimizing fatigue, so this was for upstream migration, and the simulated fish would pick a path that would minimize their accumulated fatigue. And then habitat preference, so I'm going to come back to that because that's the next chapter. There was multi and single species models, and I don't really have time to go into it, but it's good because at the end of the day, we're not wanting to pass just one species, we're wanting to try and find a solution that works for many. With IBMs, because you're simulating lots of individuals, I mean, if we look around the room, we're all quite different from each other, and you can have that in IBMs, and some of them did, but some of them didn't. So in some of these existing models, every single fish, in theory, would react the exact same way to a stimulus. Swim capacity, again, some models had, some models didn't, and it's quite important even with downstream migration because it can dictate what unfavourable flows a fish can escape from. And lastly, there's been no schooling behaviours or interactions between individuals included yet, which, as we heard from Gloria today, is really important. I will say, though, that one of the papers did talk about how you could include it, but they didn't yet. So into the future for IBM development, well, what's kind of needed? I'm not an IBM developer, right? But we need data on fish movements. We need to know what the fish are doing, how they're behaving, and for lots of different species. So what's affecting it, how does it affect it, and how different is this between different individuals? So for data, James mentioned acoustic telemetry, but I'm just going to explain that a bit more. So for those of you who aren't familiar, you have your fish, you implant an acoustic tag. This emits a unique signal with an identifier for that tag and that fish. And if you have receivers set up in your study site, the signal will be heard by them, and if you're lucky, or you get reflections. And then based on algorithms and maths and when the signal arrives at the receivers, you can triangulate the position of a given fish at a given point in time. And through this way, you can get large amounts of positional data for your fish. You can get them at very high frequencies as well, like every 1.2 seconds, for example, again, under ideal conditions. And you can analyze this data to understand movement, which is what I did in the next chapter. So this is the Altus Ried hydropower plant in southern Germany, and here barbel and grayling were tagged and tracked during their spawning migration. So we constrained tracks as well to be specifically during their known spawning migration and to represent tracks where they approach the entrance of the fish path, which is marked on the diagram. And on this I did habitat preference and movement analysis. And before I continue, I'm going to define some stuff because I am an ecologist and this is how we roll. So habitat is anything that will describe a point in space. So that could be substrate, that could be vegetation, that could be flow velocity, that could be water depth. So it could be hydraulic stuff and it can be vegetation. Habitat preference is the disproportionate usage of habitat relative to availability. Now what this means, if you consider an animal with no habitat preference and it moves through its environment, it doesn't care what there is, you'd expect over time the habitat usage to be in proportion to the availability, right? Because it has no preference. So to identify habitat preference, we need to compare usage to availability. And habitat selection is a behavior through which the fish actually selects the habitat. Yeah, sometimes I get selection and preference. There's a lot of overlap, but that's the general gist of it. And I analysed 5 step selection functions, which is what James talked about, and I'm going to go into a wee bit more detail. So step selection functions are a framework, a statistical modelling approach to model habitat preference and selection of animals as they move, movement being very important. So if we consider we have this track of a fish shown in black, so that's just some movement steps, these movement steps represent the usage. This is where the fish actually went. We can then generate some potential locations via random steps to serve as a measure of availability, and then do a statistical comparison of, okay, what differs between these black steps where the fish went and these grey steps where they didn't go, in terms of habitat but also movement attributes. So I have a box to say that. So you can analyze habitat preference and selection, and also movement characteristics and so on. For example, displacement. And when I did these, I fitted step selection functions to every single individual we had individually, so I could really focus on the individual level. The main habitat parameters that we had were flow velocity, water depth, the spatial velocity gradient, the angle of the flow, and the angle of the spatial velocity gradient. And these last two we analysed relative to the angle of the fish or a step. So it's really looking at the difference rather than the raw angle. We also had substrate data, but it just computationally didn't work for some reason. Yeah. We could also look at interactions, and this is quite important because an interaction really asked, does the fish's response to one variable depend on the value of another variable? So in this context, do we see that the fish select or prefer for faster, deeper water or faster, shallower water? Just as an example, please don't think too much about it. But we can start to investigate that. And lastly, movement. So you can have a look at displacement, and again, are they going to move faster in deeper water or faster in shallow waters? What's the general trend there? And I did a very broad model with many terms to figure out what's going on, and then did model selection to find which one fitted the data best. And because I fitted models to every individual, I then, for each species, took the average so I could look at what the general trends were. And that's what I'm going into now. So this is also being published, and I have a QR code at the end, but for general trends, barbell, we had quite a few. So in general, we saw that they preferred faster flow, like you will read in literature, which is good. Deeper water, which has also been reported in adult barbell. High spatial velocity gradient, so flow that's accelerating. And yeah, interestingly, in the direction of flow, which was a strange one, but hey, it is what it is. And then for grayling, we couldn't distinguish general trends for most terms because the individual grayling varied so much in their responses that when we took an average, it kind of just, I'll say, cancelled out to being no effect on average. And that individual variation, I think, is incredibly important. So individual variation was high overall. In barbell, we could get general trends, but there was still very high individual variation. In grayling, man, it was so high. Trust me on that. I don't have time to go into it, but boy. So just an example, this is for barbell and their selection of flow velocity. The gradient of the line represents the selection strength, or the preference. So if it's a positive gradient, it's a line going up kind of thing. The black line is the mean trend, and every grey line represents an individual. So on average, we had a positive relationship with flow velocity, and we can see that for a lot of fish that was the case, but we had 3 fish where that was not the case. 1 fish actually preferred slower velocities, and 2 fish, flow velocity had no effect. So while we have a general trend that agrees with the literature, we have individual fish who differ. And why do we have individual variation? Great question. Well, it's not related to fish size. We tested that. Fish size did not explain their preferences out of our studied fish. At the end of the day, individuals do behave differently, and we see this across animals, across a range of metrics, looking at spatial usage. You see individual variation in residency and home range and so on. But another thing could be behavioral state. So behavioral state, our next speaker will talk about a lot more, but if an animal is transiting through the environment, they may select habitat differently to if they're searching, keeping it simple. And if fish spend different proportions of time in different behaviors, then that might affect the bigger picture we get. So the work I'm doing now at the moment is sort of accounting for behavior in this way. So we've got our original data. We applied hidden Markov models as a way to distinguish behavioral states, which Jorg's going to talk about, hopefully a lot more detail. And then we get 2 states. So state 1 being resting or searching behavior, more restricted movement, and state 2 being transiting. And then to each of these data pools, apply the step selection functions separately so we can quantify habitat preference in those behavioral states. And so far, doing this improves the fits of our models ever so slightly, which is good to see. And we do get some different conclusions on a like looking at each species in terms of general trends. So, for example, in both species now, for barbel and grayling, they only really care about the depth of the water when they're in this sort of restricted movement, so when they're searching or resting. For barbel, we can now see that they have a stronger preference for flow velocity when they're perhaps searching compared to when they're just moving through the environment. And for grayling, spatial velocity gradient, they only care about that when they're searching. So yeah, and as you can see, it can tell you general trends about grayling now, which is great as well. But individual variation is still high. So while we can start to tease apart some more of the story, fish are different from each other. And another key thing here is that the resulting preferences when I compare them for barbel between state 1 and state 2, statistically they are not different in pretty much every case. So while we can do this extra work and get slightly better models, from a statistical point of view, I can't really say flow velocity has a stronger effect for searching. It does, but it's not statistically significant. Love that, don't you? So yeah, so what am I going to be doing with this? Well, the next thing I'm trying to do is evaluate these step selection functions in terms of predictability. So how well can I use the results I've got so far to predict the track of a typical fish in this system? I'm going to be doing this by a cross-validation, so having a test fish and making, yeah, you know, how it goes. And then with this, I want to use the results to parameterize an individual-based model, again, kind of like what James touched on, which I could then use to predict the spatial usage of fish at different discharges in our system, because the discharge can vary, and that will change kind of the hydraulics of the space. So it's a question of how will this change how fish use the environment? Will it make it more likely that they'll be able to find the fish pass, for example, under certain discharges compared to others? So yeah, main conclusion slide. IBMs are quite a promising tool, and they've been very successful so far in the case they've been applied to. We do need to collect more data though on more species in order to make more IBMs. Step selection functions are a very good tool to help improve understanding and sort of bridge this gap between the data we've collected and predictive modeling. And if we combine with hidden Markov models, we can account for behavioral state, and like I said, that does improve how the models fit and what we can get out of it. Individual variation is high, and I do really emphasize this a lot, as a lot of the time you see people try to put one species in a box and say, this is what this species is like, but that's not the case, and we do need to consider that individuals are not going to respond the same to the same stimuli. And lastly, as I said, I'm going to be going on to do, we can use our step selection function outputs to parameterize individual-based models, and that is the end of my thing.